Hello guys. So I hope you are enjoying with me reading Parsons. We are continuing with the chapter number 1 and this is your part 4. So we are already done with the most of the anterior segment and now we are starting with the posterior part. Let's start with the choroid. So the choroid is an extremely vascular membrane. Now this you already know. I usually give you this line also that the most vascular part of the eyeball is actually choroid. It is having the choriocapillaries, and this is also the reason why you get a reddish reflex in the retina. Otherwise, neurosensory retina is a colorless layer. It is transparent. But why do you get a orange color reflex, a yellowish reflex, or a reddish reflex? Different people say it differently because of these choriocapillaries. Um, so it is in contact. This layer is in contact everywhere with the sclera. Now we know if you look at the different layers, we have got the retina, then we have choroid, and then we have sclera. So this is retina, this is choroid, and this is sclera. So most of the places this choroid is remaining in contact with the sclera, except, okay, although not firmly adherent to it. So what happens that though we have got choroid in contact with the sclera, but actually they are not firmly adherent to it. And if you remember, we always have a space there. Usually we say that it is going into the suprachoroidal space. Even when you see the uveoscleral outflow of the aqueous humor, that time also we say that the aqueous humor is going to the suprachoroidal space. So from where this space is actually coming, it is this epichoroidal space or the suprachoroidal space that we actually are talking of, because the choroid is in contact with the sclera, but we have some space there. Like if you remember, we also have the subretinal space. So that is lying between the RPE layer and the neurosensory retina. We have a space. Similarly, they also have a space between the choroid and the sclera, and this space is called as the epichoroidal space or the suprachoroidal space. Now, if you go on the inner side of the choroid, okay, on the inner side of the choroid, we have got the basal lamina, and you have to remember, we will be talking about this basal lamina many times. This is called as the Brooks membrane of the choroid. This is called as the Brooks membrane of the choroid. So this Brooks membrane of the choroid, which is actually the basal layer of the choroid, remains in contact with the outermost layer of the retina. And what is the outermost layer of the retina? RPE layer. So basically they are trying to orient us that we have neurosensory retina, we have subretinal space, then we have retinal pigment epithelium, we have the Brooks membrane, we have choroid, we have suprachoroidal space, and we have sclera, right? Now the blood vessels of the choroid increase in size from within outward. So as they are going out and out, their size is going increasing and increasing. So that immediately beneath the membrane of the Brook, there is a capillary plexus of fenestrated vessels, the choriocapillaries. Now they are saying that choriocapillaries or the vessels are present throughout the choroid, but as you go from inner side to the outer side, the size of the vessel is increasing. So just if you look at the basal side, that is your innermost side, the innermost side will give you very small capillaries, and that is actually called as the choriocapillaries. Now upon this choriocapillaries, you will have larger size, so there will be a layer of medium sized vessels. Then as we move more outwards, then externally we will have the large vessels. Okay, so as you go out and out, the size of the vessel is increasing, nothing much. Okay, then we also have the connective tissue stroma. See, when you have vessels there, I require something which is holding these vessels together. So they are held together by the stroma, which is consisting of the pigment connective tissue cells. So, we also have the pigmentation here. Now, if you remember, when we do the naming of the different layers of the retina, then what we do? We always identify the choroid by the lumen of those large vessels. We have got something like this. So you have the lumen of large vessels, and you also see that there is pigmentation there. So we also have the pigmentation there. Why? Because we have got the pigmentation between the vessels of the choroid. So that is also one identifying feature. Then second identifying feature was your retinal pigment epithelium. These were the two things by which we actually identify that which side of the retina it is, right? So the same thing they are saying, that there is a pigmented connective tissue cells. So this is important with respect to the demarcation, that whether it is the outer side of the retina or it is the inner side of the retina. The choroid is supplied with the sensory nerve fibers from the trigeminal as well as autonomic nerves, presumably of vasomotor function. Then if we talk about the nerve supply, then it is important to know that the choroid is being supplied by two nerves. We have got trigeminal nerve as well as the autonomic nerve. So basically it is having somewhere like vasomotor function, like that is why it is having both the nerve supplies. Now the next is your posterior chamber and the vitreous humor. Posterior chamber and the vitreous humor. Now everywhere they have used a wrong word there. By posterior chamber, they mean to say the vitreous cavity or the posterior segment. So I would say that this is wrong. I will change this. This is actually the vitreous cavity. Okay. Because posterior chamber is actually present in the anterior segment itself, and that contains the aqueous humor. So vitreous cavity, I'll use the word there. It will be noticed that there is somewhat a triangular space between the back of the iris and the anterior surface of the lens. So they are saying that if you remember what was happening, it was something like this. Okay, this was your iris and this was the lens, this was your ciliary body. Okay, so this was your anterior segment, this is having anterior chamber, and this is having your posterior chamber. Now at the back we are having this vitreous cavity. Okay, now what they are trying to say, that there is a triangular space between the back of the iris and the anterior surface of the lens. So basically they are talking about this posterior chamber, this area. Okay, let me mark this area. They are saying that this is small triangular space which is lying between the iris and the anterior surface of the lens, and this space contains what? This space contains the aqueous humor. Because it is a part of anterior segment, so this space contains the aqueous humor only. So go back, having its apex at the point where the pupillary margin comes in contact with the lens. So why it is triangular? Because they are saying it is something like this. The pupillary margin, when it is coming in contact with the lens, then the shape becomes triangular. So with that respect, they are saying that it is triangular in shape, but they are rightly saying that it contains the aqueous humor. So basically I think they want to say that we will talk about posterior chamber and we'll talk about the vitreous humor, but here they have talked first about the aqueous humor. So we can cut that, because that becomes misleading, that they are saying that the posterior chamber contains the vitreous humor. So that is wrong. Now if you talk about behind the lens, behind the lens I am having a large vitreous chamber. So they have rightly used this word vitreous chamber. You can also call it as a vitreous cavity. Either we have the posterior segment or we can call it as vitreous chamber on the vitreous cavity, and this vitreous chamber actually contains the vitreous humor. Now vitreous humor, you know that in the eyeball we have got clearly two fluids. One is your aqueous humor and another is vitreous humor. Aqueous humor is water-like and the vitreous is gel-like. So vitreous is jelly-like material, and you know that it is transparent. Now though aqueous humor and vitreous humor both are transparent, but yes, consistency of both are very, very different. Aqueous humor is water-like, vitreous is, you know, glass-like. Vitreous word actually is obtained from the glass-like thing. So it is a whole gel kind of a thing which is present inside the vitreous cavity. It's very, very inert. Okay, very few cells or the leukocytes are actually present, otherwise it is very, very inert. As in other gels, the concentration of the micelle on the surface give rise to appearance of a boundary, the so-called hyaloid membrane. Hyaloid membrane, actually if you remember, I tell you this also in the diagrams, that what happens, the vitreous actually forms the false membranes, because vitreous is a gel like of a substance, so at certain places it is actually condensing and it is forming the false membranes. So wherever it is forming the false membranes, I will use a greenish color to show the membranes. Suppose this is the vitreous here, okay. So this vitreous is condensing anteriorly to form anterior hyaloid membrane, and then it is also condensing posteriorly to form the posterior hyaloid membrane. So actually these are called as the hyaloid membranes. Why actually the membranes which are formed by the vitreous are called as hyaloid membranes? Because they contain the hyaluronic acid. So that is again an important point, that within the vitreous you have something, and this is called as the hyaluronic acid. This is called as hyaluronic acid. So we have got hyaluronic acid crystals which are present in the vitreous, and that is the reason the membranes which are formed by the vitreous are your anterior hyaloid membrane and the posterior hyaloid membrane, right? Then coming to the structure of the vitreous. The vitreous body is attached anteriorly to the posterior lens surface by the ligament of the V-gut. Now what happens, if you look here, we have got a ligament. This is called as the V-gut's ligament. So if you talk of this, this is your V-gut ligament. V-gut ligament is a ligament. Sometimes it is also called as the hyaloidocapsular. It is sometimes also called as the hyaloidocapsular ligament, because it is attaching the posterior hyaloid membrane to the posterior capsule of the lens. So if you have an attachment between this posterior capsule of the lens, we have the posterior capsule of the lens and the anterior hyaloid membrane, then that ligament is called as the hyaloidocapsular ligament, and the name is also the V-gut ligament. Okay, now in the region of the aura, the vitreous cortex is firmly attached to the retina and the pars plana, and this attachment is called as vitreous base. Now there are two important things in this para. One is your V-gut's ligament and another is the vitreous base. Usually we talk about the vitreous base also. What happens that this vitreous gel-like structure at certain places, it is very, very strongly connected and attached. So what is that one place? So that is one place is vitreous base. Vitreous base is a 4 mm bend of the vitreous where it is very, very strongly attached at the ora serrata. Now what was ora serrata? If you look at the ciliary body, okay, so it is going and it is attaching to the choroid somewhere here. I'll show you in the different diagram. Suppose this is your iris and the ciliary body, and this is the choroid. So this is the uveal tract that I have tried to make, right? And inner to this you have the retina. So retina starts with a serrated margin like this, and then it is going back, and then it is continuing in the form of the optic nerve. And outer though, you know, we have got the sclera. So what happens at this area where I have ora serrata? This area is your ora serrata, the uneven margin from where the retina is starting. At this place, the vitreous is very, very strongly attached here. And if you look at the dimensions of this area, it is 4 mm band of the vitreous. So this 4 mm band of vitreous, which is very, very strongly attached to the ora serrata, this is called as the vitreous base. Now one important thing is that it is very, very strongly attached. So if there is a avulsion, of this vitreous base, then this is very much suggestive of the blunt trauma. Very, very suggestive of the blunt trauma. So whenever you are getting the avulsion of the vitreous base and you are being told about the trauma, you are having the history or any other thing suggestive of trauma, always think about the blunt trauma. All right. So this is done. Now coming to the next side, now coming to the posterior side. So posteriorly, if you see the vitreous body is connected It is connected to the margin of the optic disc and the macula, forming a ring around each structure and to the larger vessels. Now, if you remember, I told you before also that in cut section you are not able to see it, but actually eyeball is a spherical structure. So most of the places, if you see, the whole of the structure is ring-like. Now, anteriorly, it is connected to the lens, posterior surface of the lens, with the help of the V-gut ligament. Similarly, if you go posteriorly, so if I have the vitreous here, so it is attached to this optic nerve. It is attached to this optic nerve. Optic nerve is present nasally and slightly temporally I will have macula. So it is also having the dense attachment to the optic disc as well as macula, right? Both the places, and they are forming like ring-like structure around these places. So we know about the vitreous base, we know about the optic disc and the macular attachments. The primary vitreous is concentrated into the center of the globe by the secondary vitreous and forms the canal of Clockway, which contains material less dense than the secondary vitreous. Now they are talking about the basic structure. So what happens if you start with the intrauterine life? Okay, if you start with the intrauterine life, actually what happens, whole of the vitreous in the intrauterine life is the primary vitreous. Why? Because it is supplied by a artery which is called as a hyaloid artery. Which is supplied by artery called as a hyaloid artery. Now what happens, around 3rd to 4th week of the intrauterine life, there is starting of regression of this hyaloid artery, and therefore now we have to change this primary vitreous to the secondary vitreous. So now what happens, it starts concentrating inside a canal. So now the primary vitreous starts compressing and it remains only inside this canal. This is your primary vitreous, which was also having an artery inside it. This one, this is your hyaloid artery, while the rest of the vitreous that is following here, that comes the secondary vitreous. Rest of the vitreous is your secondary vitreous. So this will be your secondary vitreous. So what, that is the thing they are trying to say, that slowly and gradually what is happening, that whole of the vitreous cavity starts filling with the secondary vitreous, and primary vitreous is only concentrating inside a canal. And what was the name of this canal? The canal of Clockway. And the name of this was the canal of the Clockway. So canal of Clockway is the area which is extending from, if you see, from the posterior surface of the lens, and then it is going up to the optic disc. It is also going up to the optic disc here. And if you see, actually it remains after birth or not, that is very, very doubtful. So this canal of Clockway, which is containing this hyaloid artery, so if at all you are getting the primary vitreous after the birth, then it is found inside this canal. Now whether you have this canal or not after the birth, that is very, very doubtful, but sometimes sometimes you have that persistence of the primary vitreous. There you can have this canal of Clockway along with the hyaloid artery, but otherwise it exists or not, that is not known. And this is actually known to be associated with the Patau syndrome. This is known to be associated with the Patau syndrome. All right. Now coming, the body of the vitreous. If I talk about the body of the vitreous has a large and loose fibrous framework of the collagenous fibers, whereas its cortex is made up of collagen-like fibers and protein. So they are saying that there are two parts of the vitreous. One is your body and another is your cortex. So the body is made up of loose tissue, while if I see the cortex, then that is made up of collagen-like fibers and protein. So somewhere, you know, the composition of the vitreous is not same at every place. So somewhere it is loose, somewhere it is more dense. So they are trying to tell that only. Now we come to the retina. Retina corresponds in extent to the choroid. So if you look at the extent of the retina, then it is same as that of the choroid, which it lines, although the same embryological structure is continued forward as a double layer of epithelium as far as the pupillary margin. So, you know, when we talk about the extent of the choroid and the retina, that same ciliary body, okay, after this we were having both the layers, we are having this retina also, and then we have got the choroid also, both of them are there. And they are saying that the same neurological structure is continuing as a double layer of epithelium as far as your pupillary margin is also concerned. So if you see here, this iris is also there. So as far as your pupillary margin is concerned, because if you remember this, we had talked in the earlier form that if you start with the neuroectoderm, so this neuroectoderm was forming the optic cup. Do you remember? An optic cup was having the two layers, the outer as well as the inner layer. It was forming the two layers, outer layer and the inner layer. Outer layer was forming the pigment epithelium. This was forming the pigment epithelium of the retina, then of the ciliary body as well as the iris. So this is continuing, and then inner one was forming the neurosensory retina. So that is the same thing that they are trying to say, that we have a double layer of epithelium as far as the pupillary margin. So if the two layers of the epithelium are traced backwards, the anterior layer in the iris is found to be continuous with the outer layer of the ciliary body, and this again is continued with the pigment epithelium of the retina. So same thing I told you, that the pigment epithelium of the retina, ciliary body and the iris, they are continuous, and that is why it is said that the epithelium of the conjunctiva, cornea, the eyelids, the lacrimal apparatus, all these are coming from the surface ectoderm, but the epithelium of the retina, of the ciliary body and the iris, they are coming from the neuroectoderm. They are coming from neuroectoderm. Even neuroectoderm is also giving rise to the iris muscles. It is also giving rise to the iris muscles. This is also an important question that they are usually asking. Now, okay, then they are saying that it is also continuing with the single layer of hexagonal cells which are lying immediately adjacent to the membrane of the brush. So what is actually the pigment epithelium? The pigment epithelium of the retina is a single layer of hexagonal cells, because you know RPE layer is the outermost layer of the retina, and that is continuous with the basal lamina of the choroid, that is your Bruch's membrane. Similarly, the posterior layer of the iris is, although pigmented, passes into the inner unpigmented layer of the ciliary body, and this suddenly changes at ora serrata into highly complex visual retina. Similarly, they are saying that anteriorly this pigment epithelium is also continuing into the iris. There also we have a pigment epithelium. It is also going into the ciliary body, and suddenly it changes at the ora serrata, because ora serrata is the place where the retina will start. Right. Now the retina consists of a number of layers formed by three strata of the cells as well as their synapses. Now we are talking about the type of cells which are present in the retina. We have got three type of cells. We have got the visual cells which will be there in the visual cycle, obviously will be present on the external aspect. Then you have got a relay of bipolar cells. Bipolar cells are present in the intermediate one, and then you also have the ganglionic cells, and ganglionic cells are the axons which run into the central nervous system, because you know it is actually the nerve fiber layer which is continuing to form the optic nerve, and nerve fiber layer contains the axons of the ganglionic cells. So we have the visual cells, the rods and cones, photoreceptors present on the outer side, we have bipolar cells present in the middle, and we have got the axons of ganglionic cells present towards the inner side, and the axons of these ganglionic cells are continuing as the optic nerve towards the central nervous system. Now we start with the layers of the retina. Now start with the layers of the retina. So we will start from the outer side and we will go towards the inner side. So they have started with the rods and cones because they have already talked about the retinal pigment epithelium. So see, they are saying that most externally in contact with the retinal pigment epithelium is a neural epithelium, the rods and cones which are the end organs of the vision. So obviously the outermost layer is not the rods and cones, it is the retinal pigment epithelium which always remain in contact with the Bruch's membrane of the choroid, and after that we have got the layer of photoreceptors, the rods and cones, and these are considered to be the end organ of the vision. The microanatomy of the rods and cones reveals the transductive region. This is your outer segment. Now this is a very, very important thing. We will be seeing this in successive chapters, that there is a thing which is called as the phototransduction. there is a thing which is called as the phototransduction. Phototransduction is a series of visual and complex events which are taking place in the visual cycle, which is converting the light energy into the electrical energy. So this is actually occurring in the outer segment of the rods and cones. So you have to remember this. This could be asked as a direct MCQ also, that it is a phototransductive region. So phototransduction is actually running in the outer segment. Then we have got a region for the maintenance of the cellular homeostasis. So what is actually, if you look at the structure of the rod photoreceptors, see this, I'll try to show you, then you'll get to understand. See, this is a photoreceptor. So photoreceptor, this is your RPE, and then starts your photoreceptor. Now this consists of an outer segment, then an inner segment, then we have got the nucleus, and this area will be your synaptic body. This area will be your synaptic body, something like this. So it is in this outer segment that we will have the phototransduction. In this area, we will have the phototransduction. In the inner segment, we will have the maintenance of the homeostasis. Here, you will have the maintenance of homeostasis. Nucleus will be present in the outer nuclear layer. And then you have the synaptic body. This is present in the outer plexiform. In the outer plexiform layer. So this is how we are getting oriented, what is happening after what, and how the layers are oriented. So in this way, I have already seen, we had the RPE layer, then we have seen the layer of the rods and cones, then their nuclei going, okay, nuclei will be going into the outer nuclear layer, but before that we have an internal limiting membrane or internal limiting layer, and then you have the outer nuclear layer and the outer plexiform layer. So up till this we have seen. So see, they have said that this inner segment is helpful in maintenance of the homeostasis. Then they are saying the nuclear region or outer nuclear layer and a transmissive region outer plexiform layer. Now believe me or not, these have been asked so many times as a question, especially names. I don't know what will happen in INI CET exam, because now most of the Central Institute examinations have been merged, but previously AIMS people were lifting the questions from the Parsons lines. So nuclear, and this is a question 3 to 4 years back, that where do you have the nuclei of rods and cones? So you have this in the outer nuclear layer, and where do you have the synaptic body? That is in the outer plexiform layer, right? Now when the outer segments of the rods are sectioned parallel to their long axis, they are seen by the electron microscopy. Another important thing is that which microscope will be used to see them? It is the electron microscope, to consist of a boundary or a cell membrane which encloses a stack of the membrane systems. The disc in the rods of many species are continuously renewed. New discs are formed in the region of inner segment, and they are progressively displaced towards the pigment epithelium. Rod discs have a limited life and eventually they are lost. So this is nothing significant. Okay. So if you look here, now the thing that I was telling you, that the choroid will show you these lumen. So lumen is present in the vessels. So this is your lumen of the vessels. This is a very, very important identifying feature whenever I want to get that which area is the outer layer out towards the outer side of the retina and which is towards the inner side. So I will always use this. So if I know that this is the lumen of the vessels, that means this area is a choroid, and immediate to the choroid I will have single layer of hexagonal cells. So this layer will be RPE layer. After the RPE layer, I have rods and cones, and see this area. If you see this area, you will be able to see very, very long segments. So this is your outer segment and inner segments which are actually present inside the layer of rods and cones. See, this is your photoreceptor epithelium. Then the next, where you will have dark nuclei kind of a thing. So I'll try to show you, see this, you are having this nuclei, darker nuclei, they are present. So these are actually, is this visible? So these are actually the nuclei of rods and cones. These are nuclei of rods and cones. So see, this is your outer nuclear layer, and ultimately they are synapsing. So here you have the synapse. Synapse means, okay, sorry, ultimately you are synapsing here. So this is your synapse. So this will be your outer plexiform layer. Now again you have an area where you are able to see the pigmentation, darker one, nuclei area. So this is also nuclei, but this is towards the inner side. So I will say this is your inner nuclear layer because this is towards the inner side, right? And again they will be synapsing. So I will have the inner plexiform layer. So we have reached up till here. First we'll try to read and then we'll come back to this diagram. Now at the junction of, see they are saying something. They are saying that if you look at the junction of the inner segment and outer segment, the cell body is like constricting, and we were looking at them by the electron microscopy. So electron microscope reveals a connecting cilium which is always eccentric and provides the only link between the inner and outer segment. So they can ask you one question, that what is the connecting link between the outer segment and the inner segment? It is the cilium. And how can you see it? With the help of the electron microscope, you can see the connecting cilium between the outer segment and the inner segment. Then if I talk about the retinal pigment epithelium, like they have first talked about the rods and cones, and after that they are talking about the retinal pigment epithelium. It consists of a single layer of hexagonal cells. Very, very important. This is lying between the photoreceptor outer segment and the Bruch's membrane. So actually this RPE layer is lying outside. It is the outermost layer of the retina. They assist in the metabolism of the retina by transporting the selected substances to the receptor cells. Then second important thing, first important thing is where it is present. Number two, what is its function? It is mainly helping in the metabolism because it is helping the transported the selected substances to the receptor cells. Products of metabolism are freely exchanged between these receptor cells and the pigment epithelium they are giving. Now the most striking inclusions in the pigment epithelium are the organelles. Now most important thing, why I am saying that it is a pigment epithelium, because it contains the pigments. So there are certain organelles which are responsible for its color, that is important. And what is that? Melanin granules. So because it contains a pigment melanin, see, if you remember, I told you that when chloroquine toxicity is taking place in the retina, we have bullseye maculopathy. So the first thing that it was doing is it was combining with the melanin pigment of the RPE layer. So we have the presence of the melanin granules in the RPE, and then it was causing the disruption at the junction of outer segment, inner segment. So it was most probably it is breaking that cilium which is present at the junction of outer segment, inner segment. Now most of the light which is passing through the retina is not absorbed by the pigments which are there in the photoreceptors, and that is absorbed by these granules. What they are saying, that when light is passing through the retina and it is not absorbed by photopigments in the photoreceptor outer segments, it is absorbed by these granules. So when the light is coming, okay, it is first going to the neurosensory retina, then it is going to the rods and cones, and then it is going to the retinal pigment epithelium. So even those light which is not absorbed by these photoreceptors, even that will be absorbed by these melanin granules which are present in the pigment granules. And I think this is important, and that is why they have kept RPE after that, because otherwise the light would not have reached the photoreceptors. Then another important thing is your phagosomes. Phagosomes are known to be discarded rod discs that have been engulfed by the pigment epithelium. So sometimes you may get a question on these phagosomes also. What are phagosomes? They are nothing but the discarded rod discs which are actually engulfed, which are taken up by these pigment granules. Okay, the phagocytic capacity of this pigment epithelium is demonstrated in the response of retina to the injury. So this is shown whenever there is injury to the RPE by the laser IR radiation, when the number of the phagosomes increase significantly. Whenever there is a trauma, it has been seen that the number of phagosomes suddenly starts increasing. So that shows that it is undergoing the phagocytic activity. Then the next is the outer nuclear layer, which contains the nuclei of rods and cones. I told you this is a direct aims question. Then you have the outer plexiform layer, which contains the synapses of the rods and cones. So that can again be asked. Then you have inner nuclear layer. Now nuclear layer means again it contains the nuclei. So nuclei of which cells? Here they have used just the word bipolar cells, but actually it contains many other cells also. So usually I use the word hub. It contains the hub of the cells. H for horizontal cells are also there, and A for amacrine cells. A for amacrine cells, and B they have already given bipolar. But if you have to choose one, you have to choose the bipolar cells because now they are the second order neurons. Then we have inner plexiforms. So again, this is synaptic. Next is your ganglionic cell layer. Ganglionic cell layer is again important because the axons of the ganglionic cells are running into this nerve fiber layer, which is continuing as optic nerve. Now again, this is very, very important. If you remember, I always tell you in the classes that optic neuropathy consists of the death of ganglionic cells. Why optic neuropathy contains the death of ganglionic cells? The reason is that optic nerve is made by the nerve fiber layer, and nerve fiber layer contains the axons of ganglionic cells. That is why whenever we have optic neuropathy, we have death of the ganglionic cells. So with this, we have completed the choroid in the retina. I think this is all for today. This is quite complex. We have solved many questions and MCQs today. I have discussed so many AIMS questions today, and an important task for you people, you have to work on the layers of the retina. You make a flow chart, make a table in your notebook and try to write all the layers. See if you are able to write them in the sequence, right in front of it, what is the peculiarity or what it contains, because definitely it is going to give you a question, and try to see if you are able to decode the histology of the retina. That is again very, very important. You could be asked any of the layers of retina. See you in the next part of this series. Thank you and happy ophthalmology.